சிஸ்டம்ல பார்க்குறது இங்கே வராதா சார்? இப்படியா? நான் போய் ஸ்டார்ட் பண்ணலாமா? Hello students, good morning. Welcome to Future Doctor Academy. So yesterday we saw about the cell, the unit of life, the introductions and the cell theory, who invented cell theory, and we saw about overview of a cell, right? Okay. So now before starting the class, because today the chapter is prokaryotic cell. In cell, the unit of life, we are going to see about prokaryotic cells. So before we enter into the topic, so we'll recollect the previous one. Okay. Who proposed living cell? So without seeing your book, you should answer. Who proposed? Is the first discovery in your subject, right? Anton von Leeuwenhoek, right? Okay. And then who is the botanist who invented plant tissue that is having in cells? Matthias Kleeden. Matthias Kleeden in 1835. Okay, fine. And who is that zoologist? Theodore Swan. Okay. In 18, that is 1836, and this is 18. Yes, tell me. So 1839. Okay. 1838 and 1839. And who discovered who gave us final shape to our, cell? That is, cell is dividing from the pre-existing cells. Who proposed? Rudolf Virchow. So in which year? 1855. Okay. So these are all the cell theory who invented, we have seen yesterday, right? So and then we have an overview of cell. So what you have learned in the overview of cell? So inside the cell, what will be there? Yes, very good. Very good. Okay. Okay. So parts present inside the cell, what we call? Organelles. Organelles. So organelles is parts present inside the cell. Okay. Then which is the smallest cell? Smallest cell, and what will be the length? Okay, mycoplasma 0.3 mu meter. Okay, that is, mu meter is very important in length. And which is the largest single cell, isolated largest single cell? That is an egg. So yes, ostrich. Very good, very good. So and then in human red blood cells, what will be the diameter? 7.0 mu meter in diameter. Okay, right. Okay. So these are all the things we saw yesterday, and today we are going to see about prokaryotic cells. So what is prokaryotic cells? What is prokaryotic cells? So before also we have cells having different shapes and size, no? What are all the shapes? Okay, very good. That is cuboidal. Then columnar, polygonal, disc-like. So for disc-like, we have an example of hemoglobin, right? RBC we have shown. So like that, in prokaryotic cell, what is prokaryotic cell? Prokaryotes means what? So it is having a nucleus, eukaryotic having troop nucleus. This is absence of nucleus, prokaryotes, right? So here we are going to learn line by line, okay, in NCERT. So the prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO, that is pleuropneumonia-like organisms. So prokaryotic cells, that is absence of nucleus, we already know. So it represents like bacteria. Bacteria is one type of prokaryotic. Apart from bacteria, what are all the things in prokaryotes? Apart from bacteria, give one example for prokaryotic cell. Yeast, very good. Okay, yeast, amoeba, these are all the things. So, and blue-green algae. What is blue-green algae? What is blue-green algae? Blue-green algae is otherwise called as, tell me, blue-green algae. Here in your NCERT, blue-green algae is there. So what is blue-green algae? This it is giving a color, right? So blue color and green color. So what will be the other name for blue-green algae? This is a bacteria. This is a bacteria. Organism, living organism, so bacteria. What will be the other name for blue-green algae? What is? Cyanobacteria, because it is giving color, right? So that is blue-green algae. And like mycoplasma and PPLO. What is PPLO? Mycoplasma is a bacteria. It is the smallest bacteria, which is 0.3 μm in length. We already seen. And what is PPLO? PPLO is an pleuropneumonia-like organisms. So pleuropneumonia means it is an organism. It is also an bacteria that giving rise to pneumonial fever, pneumonia. If anyone is presenting from pneumonia fever, pneumonia and all, this organism, this bacteria will be there, PPLO, that is pleuropneumonia-like organisms. So they are generally smaller. Which are generally smaller? These organisms, that is bacteria, blue-green algae, mycoplasma, PPLO. These are all the organisms which it's generally very small in shape, because it is the smallest bacteria, right? And multiply more rapidly, even though if they are small, but it gives rise to a new one, new young ones more rapidly. Okay. Then the eukaryotic cells. So comparing to eukaryotic, prokaryotic cell organisms will be grown faster, grown rapidly. Okay. Then they may vary greatly in shape. This is also this prokaryotic cells also varies from, it is having a different shape, size and all, because every single cell is having a different shapes and size. We already know, we already studied also, and it is having a unique character. Comparing to including prokaryotic and eukaryotic, they both are having unique character. So here in prokaryotic we have 4 basic shapes of bacteria. So we have going through the shapes of bacteria. So what will be the shapes without seeing your book and systems and all? So what will be the shapes? Give some 2 shapes. What? Okay, irregular shape, that is we have already seen in an overview of cell, because I am asking about prokaryotic cells. Okay, rod like. Then spherical shape. Okay, so here we are going to see. So it may varies from size and shapes. The 4 basic shapes of bacteria are bacillus. So bacillus in the sense what? Rod like. So like this. like this will be the shape. And then second one is coccus, that is spherical in shape. Coccus, that is spherical in shape. This is rod-like, and this is spherical. And next one is what? Next one is what? Vibrio. Vibrio means comma shape, right? So what is comma? This is, we have already, we'll put comma in this type, no? So this is an vibrio shape, comma shape. And next, fourth one is spirulum. Spirulum means spiral shape. This is a spiral, no? You are seeing a spiral book like this. This is a spiral. So, in prokaryotic cell, there are 4 basic shapes. What are they is, first one is bacillus, rod-like shape, bacillus this is. And next one is coccus, that is spherical in shape. And third one is vibrio, that is comma shape. And last but not the least, fourth one is spirulum, that is spiral in shape. So, these are all the 4 basic types of prokaryotic cells. And then we will move on. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. Okay, some organisms of we are seeing about prokaryotic cells, they are fundamentally similar. Fundamentally they are similar, but there are some various types of shapes and size, but it is having an function. Every single cells in prokaryotic cells, that is comma shape, spherical, spiral, rod-like. So these are all having a similar and like functions and all it is having. And then next one. All prokaryotes have a cell wall. We already seen yesterday that animal cell and plant cell. Cell wall is there. This is an cell wall. So, all prokaryotes have a cell wall surrounding the cell membrane. All prokaryotic cells was covered by cell wall and surrounding by cell membrane. And cell membrane except mycoplasma, but in animal cell, in plant cell, mycoplasma is absent, whereas animal cell, it is present. Okay. The fluid matrix, what is fluid matrix filling? What is fluid matrix? Yesterday we saw. Fluid matrix like substance. Cytoplasm. Fluid matrix like substance, we have already seen. Right. Okay. Fluid matrix substance, that is fluid filled area covered by the, yesterday we already seen, no? Area which is nucleus is there. So area means a ground. So what is the ground? Which organism, which part of the organ is ground? Cytoplasm. So that is a fluid matrix area. Okay. So the fluid matrix filling the cell is the cytoplasm, because this cytoplasm is contained full of fluid matrix, and there is no well defined nucleus. So in prokaryotic cells, not a nucleus, well defined nucleus. In prokaryotic cells, there is no nucleus, absence of nucleus. And then it is a lack of membrane bound organelle in prokaryotic. So there is no well defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. So here in nucleus only we are having a genetic material. We already know that is on DNA, RNA. So these are all the genetic material present inside the nucleus. But in prokaryotic cells, there is no well defined nucleus. So where will be the genetic material? Where will be the genetic material? So it is having a single cell. So everything will be inside. Okay. So there is, we should not basically seen in genetic material in the prokaryotic cells. So, in addition to the genomic DNA, that is the clear single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. So, what is that? It is telling some bacteria is having a single chromosome, circular DNA and smaller DNA. So, in this we are going to see some bacteria, just a minute. So, see, imagine this is a bacteria. So, this is an small circular DNA in a single prokaryotic cell. So, it is a small circular DNA and this is genomic DNA or plasmids. Okay. So, see, imagine in this point we have seen DNA, these smaller DNA are called plasmids. So, many bacteria have many bacteria. So many bacteria is having small circular DNA. This is small circular DNA and what is outside the genomic DNA. So, this is genomic DNA, it contains DNA genetic material and some DNA is there, that is small circular DNA. This genomic DNA is otherwise called as plasmids. Okay. So, these smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters. So, this plasmid contains some phenotypic characters. What will be the phenotypic characters? What will be the phenotypic characters? Phenotype. So, expressing character, express. So, what are all the character that is expressing is called an phenotype. Okay. So, phenotypic characters. Phenotypic characters to such bacteria. So, some bacteria is giving an phenotypic characters. So, next one such character is resistance to antibiotics. So, this character, expressing character, no. So, we are having an character in human beings, we are expressing some characters, that is police force our body, that is a WBC, because it is an resistance to an antibiotic. So, same way in prokaryotic cells, it is also having an resistance power that is giving rise to antibiotics. And then in higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. So, later on you are going to study in the higher classes that DNA, what will be this DNA is going to use. So, which transformation will be going on to the DNA. So, you will be studying in your higher studies. So, nuclear membrane is found in eukaryotic. So, here in bacteria, nuclear membrane, there is no nucleus. So, in one cell it is having everything, but in eukaryotic nucleus is present. And then no organelles like the one is eukaryotic are found in the prokaryotic cell except for ribosomes. We have seen yesterday itself. So, ribosome is present both in eukaryotic as well as in the prokaryotic cell. So, except a ribosome, some other organelles, some other parts will be present in prokaryotic where it is absent in the eukaryotic cell. So, in eukaryotic cell some organelles will be present, but for example nucleus. So, nucleus will be present in the eukaryotic cells and nucleus will be absent in the prokaryotic cells. For example, I have stated. And then prokaryotes have something unique in the form of inclusions. So, prokaryotic cells having some unique form, something unique. So, some person will be have a unique character. no other one will be having, no. So in prokaryotic cell we will see the unique function that is excluding inclusions. So what is inclusions? What will be the unique inclusions that a prokaryotic cell is having? That is a phosphate and then granules. These are all the inclusions. Inclusions means these are all the things prokaryotic cell is having. And then a specialized differentiated form of cell membrane called mesosome, that mesosome is the characteristics of prokaryotes. So specialized differentiated, the cell membrane is present, right? In prokaryotic cell membrane is present. So it is an specialized cell differentiated, that is called an mesosome. So mesosome will be like this. So mesosome will be like this. So mesosome is an infolding structure. This is an infolding structure. Mesosome is the character found in the cell membrane, but it is having a unique character. So what is the unique character? It is essentially found infolding cell membrane. So this is the infolding. Infolding. This is infolding. Sorry. So this is infolding. So it is very essential, it is present in the cell membrane, it is having an essential character, a unique character, that is an infolding cell membrane. So this is an infolding. So in this infolding we have so many types of infoldings, cell membranes. So we are going to see one by one. So next we are going to see about an cell envelope and its modification. So far any doubt, students? Okay. Now we are going to see about a cell envelope and its modification. So what is cell envelope? What is cell envelope? What is cell envelope? Tell me. Cell envelope means what? Cell envelope. Yes. What? Okay, covering layer, that is cell envelope. And how will be the modification? Some modifications will be there in the cell. So we are going to see about that. So most prokaryotic cells, particularly the bacterial cells. So practically we know that prokaryotic is an example for bacteria. So in particularly here in your NCERT, it is saying that bacterial cells have a chemically complex cell envelope. Some envelope will be surrounding, envelope means cover. So cover, some envelope is present in the bacteria. So the cell envelope consists of a tightly bound 3-layered structure. So one cell is here, a cell. So in this cell a tightly bound layer is enveloped. Okay, so tightly bound 3-layer. How many layers? 3 layers. So 3 layers in the sense. So here so these are all the 3 layers. So in this 3 layers, tightly bound 3 layers. So that is the outermost glycocalyx. So in this 3 layer we have seen, so first layer, second layer and third layer. So which will be the first outermost glycocalyx? Which layer? Or first, second or third? First. This is an glycocalyx, outermost glycocalyx. And then next glycocalyx, followed by the cell wall and then the plasma membrane. So glycocalyx is followed by which organ? Cell wall, and then cell wall and then plasma membrane. Okay. So in this prokaryotic cell, especially we are going to see, the bacterial cell. So it is having an envelope. In this envelope, that there is an tightly bound 3 layers are present. Here you can see, sit here. No issues. So we are seeing a 3 bounded layers. In 3 bounded layers, that is first layer is, outermost layer is glycocalyx. So after glycocalyx, it is followed by an which layer? Cell wall and then last plasma membrane. So first layer, outermost layer is glycocalyx, second one is cell wall and third one is plasma membrane, that is cell membrane. Cell membrane, plasma membrane, both are same. Although each layer of the envelope performs distinct function. So each layer like glycocalyx, cell wall, plasma membrane, so each and every layer is having different functions. So we are going to see the different functions. They act together as a single protective unit. This 3, that is glycocalyx, cell wall and plasma membrane, these 3 layers are uniquely formed and act as a protective unit, because it is giving the whole, this glycocalyx, cell wall and plasma membrane protect the cell. This 3 layer are protecting a cell. So that is an unique function. And the next, bacteria can be classified into 2 groups of the basis of the difference in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram-Wiss. Though that take up the Gram stain are Gram positive and the other that do not are called Gram negative bacteria. So what is Gram? What is Gram? So yeah, he is a scientist. So because he was invented, he proposed that bacteria will be, because he how the scientist will be discovered? Which experiment? What experiment will be done? So some experiment he is done through a staining dye procedure. If it is giving a color, it is positive or negative. So for positive, which color will be present? For negative, which color will be present? Okay. So staining here, staining is color. So positive means which color? Gram positive means? Gram positive is violet color. Gram positive is here. Gram is the name of the scientist. So he only proposed that whether it is Gram positive or Gram negative. So if it is Gram positive means it is violet in color. If it is Gram negative means a red in color. Okay, red in color. These are all the difference in your NCERT. It will not be given in your NCERT. Okay. So some staining procedure will be done through bacterial, because a pinch of bacteria will be taken from and it will be examined through the microscope, electron microscope, using an staining method, that is dye method. So it will be giving some colors. If the color gives a violet color, that is a Gram positive. For example, we have undergone some E. coli test, because we will have some UTI problems and all, because all of them are having using a common toilet. So a common bacteria is E. coli bacteria. So if we will study through a staining procedure, that is solution will be there for an urine test. So here that strip will be stepping down into the urine test, means if it is color is changed to violet or red, means that will be the Gram positive or Gram negative. It will Gram positive E. coli, Gram negative E. coli. That reports will be shown, that like results. So this will be the Gram positive and Gram negative. And glycocalyx differs in composition and thickness among different bacteria. So glycocalyx is differ from some compositions. So glycocalyx is having some compositions. So we are going to see about what are all the compositions. Just two compositions that glycocalyx is having. So it could be a loose sheath or tight sheath, rough tight, thick and rough tight. So loose sheath is called an slime layer. Here this glycocalyx so here this glycocalyx is having an two composition. What are they means? First one is loose sheath. Second one is thick and tough layer. So loose sheath is called what? Thick and tough layer is called what? Loose sheath is slime layer. You have seen the slime, right? That is a slime layer. And thick and tough called capsule. So glycocalyx is having an two composition. One is a loose sheath, that is a slime layer, and second one is thick and tough, that is called an capsule layer. Okay, and cell wall. So first thing we have seen, so glycocalyx is the outermost layer and it is having an two composition, that is a loose sheath and an thick and rough. So the loose sheath is called slime layer and the thick and rough sheath is called capsules. So next one we are going to see is cell wall. So what is cell wall? Cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. So cell wall, here second one is innermost layer, sorry, middle layer is cell wall. So cell wall is giving what? Cell wall is giving what? Shape. It gives the shape. Cell wall is giving the shape to the bacteria, or even not only in bacteria, not only in prokaryotic, even eukaryotic also cell wall is giving the shape. So it is giving the shape and structures, and this cell wall protects the cells. Okay. So protects the cells and it gives the structural support to the cell. So this cell wall also preventing the cell from the bursting. What is bursting? Bursting means what? Collapsing means what? பரவாயில்ல தமிழ்லே okay, no issues. ஓகே, கொலாப்ஸ் ஆகிடுது. இப்போ ஒரு இடத்துல இருக்குது அப்படின்னா அது அப்படியே கொலாப்ஸ் ஆகிறது நம்ம கொலாப்சிங் அப்படின்னு சொல்கிறோம். தென் வாட் இஸ் பர்ஸ்டிங்? பர்ஸ்ட் அவுட், வெடிக்கிறது. ஒரு இப்போ நம்ம ஒரு செடி எடுத்துருக்குறோம். பிகாஸ் வி ஹாவ் சீன் இந்த ரெஸ்பிரேஷன் அண்ட் பிளான்ஸில் நம்ம எல்லாம் பார்க்குறோம் அப்படின்னா, அப்போ ஃப்ளவரிங் பிளான்ஸ்லாம் பார்க்குறோன்னா அப்போது அந்த போலன் கிரெயின் என்ன ஆகுது? பர்ஸ்ட் ஆகுது. ஸோ அந்த போலன் பர்ஸ்ட் ஆனால் தான் உள்ளேருந்து போலன் கிரெயின்ஸ் எல்லாம் வெளியில் வரும் இல்லையா? ஸோ அப்போ அது பர்ஸ்ட் ஆகுது. ஸோ அந்த மாதிரி பர்ஸ்ட் ஆகாமல் பார்த்துக்கிறதுக்கும் அண்ட் தென் கொலாப்ஸ் ஆகாமல் பார்த்துக்கிறதுக்கும் திஸ் செல் வால் இஸ் prevent பண்ணுது. Okay, and the next one is the plasma membrane is selectively permeable in nature. That what is plasma membrane? This is a cell membrane. Cell membrane is a plasma membrane. First we have seen cell wall, we have seen, and then plasma membrane. Plasma membrane is selectively permeable in nature and interacts with the outside world. So outside world, this plasma membrane is an permeable in nature, permeable very closely, because it is giving some functions and all. So it is permeable in nature and interacts with the outside world. So this membrane is similar structurally to that of eukaryotic. So this plasma membrane, what this plasma membrane is going to see? It is similar. It is giving a similar structure to the eukaryotic cells, this plasma membrane. And last one we are going to see is a specialized membrane, a special membranous structure of the mesosome. So we have seen that mesosome. What is mesosome? we have seen, no? What is mesosome? Here we have seen. What is mesosome? What is this layer? Just now we have seen. What is mesosome? An infolding structure. Infolding, we have seen, no? So this is an mesosome. So here mesosome is having a different modification. Some modifications will be there. So what will be the modification? Mesosome, which is formed by the extensions of plasma membrane. Here this is an plasma membrane. So here it is having some mesosome. So with, what will be the extensions? What will be the extensions we are going to see? So these extensions are in the form of vesicles. First one vesicles, second tubules, and third one is lamella. So vesicles means what? And then tubules means what? Lamella means what? We are going to see. So vesicles na eppadi irukum? Eppadi irukum? Mesosomes அப்படினா it is an infolding layer அப்படி நம்ம சொல்லிட்டோம். So and then what will be the, that it is having some modification, that is vesicles, tubules and what? Third one, lamellae. So what will be the extensions? What will be the extensions? So here vesicles. First thing, vesicles. So vesicles means so this is vesicle, and next one is tubules. So this is tubules, and third one is lamellae. This is the structures of vesicles, tubules and lamellae. Okay. Can you all able to see the board? Okay. So and then next this is the mesosomes. This is the mesosome. So mesosome is having some modification, that is having and some extensions of plasma membrane. So that is vesicles, tubules and lamellae. Vesicle is this shape. The infolding will be here. Some mesosome is called an infolding structures. The cell membrane's infolding structure is called mesosomes. So this mesosome is having similar structures. So this is an vesicle structure and tubule structure, and this is an lamellae. It is an irregular shape. Yeah? So this, and then they helps in cell wall formation. Which helps in cell wall formation? This mesosomes. Any modifications of this mesosomes will help the cell wall. So it first thing, cell wall. It helps the cell wall to formation what? To second one is first one is help the cell wall formation. Second one, DNA replication and distribution of daughter cells. So first thing is help the cell wall to formation. Second one is DNA replication. DNA replication we will study in the cell divisions. So in S phase DNA will be replicated. If I am telling that in which phase DNA will be replicated? S phase. So what will be the S phase? We are going to study in the cell division. But in the cell division I will ask question, in which phase DNA will be replicated. You should answer the questions. Okay. So DNA replication, and last one is distribution of daughter cells. Distribution to so these are all the functions it is having, that mesosomes. Okay. So first one is help cell wall formation, second DNA replication, third one is distribution to daughter cells, and then Next, they also helps in a respiration segregation process to increase the surface area of the plasma membrane and enzymatic content. So, what is also help this mesosome is helpful for respiration secretion process. Some enzymes will be secrete inside. So, enzymatic content means what? What will be the enzymatic content? So, respiration. So, here we will having some enzymatic content. Some cells or some in everything, each and everything is consuming some enzymes. So, one single cell is having some enzymatic consistence. So, you will study in the respiration in plants in glycolysis. So, some 10 steps will be there in the glycolysis, you know. So, here the 10 steps we are having some enzymatic conditions. So, some enzyme will produce like that if the enzyme is produced, ATP will be given for. So, likewise some enzymes, each and every cell is producing some enzymes, that is an enzymatic content. For example, respiration. In some prokaryotes like cyanobacteria is blue-green algae that are other membranous extension into the plasma called chromatophores which contain pigments. So, chromatophores, what is chromatophores? So, this chromatophores is helpful for the pigment colorization, that is an blue-green algae. Bacterial cell may be motile or non-motile. So, what is motile and what is non-motile? If bacterial cell is motile and non-motile. So, what is motile and what is non-motile? Motile means what? Non-motile means what? Motile means movable, non-motile means not movable. They can't able to move. So, what will be the give some example for motile and non-motile? In your NEET examination, this point will be asked for several times. So, bacteria may be motile or non-motile. So, motile means flagella. Flagella, it is giving an peristaltic movement, like this it is moving, right. So, flagella is one good example for motile condition. So, that is movement. If motile, they have thin filamentous extension from their cell wall called flagella. So, flagella. So, motile means it is having flagella. If non-motile means flagella will not be extended. Bacteria shows a range in the number of arrangement of flagella. So, bacteria will be showing some flagella. So, we are going to see the flagella parts. So, bacterial flagellum is composed of three parts. What are all the three parts present inside the bacteria? Filament, hook and basal body. So, here so, see, this is an bacteria. First thing, what will be the three parts? Filament, hook and basal body. So, filament means what? So, this is filament, and because filament is moving, extending from the bacteria. So, this is hook and this is basal body. This will be the structures, and the filament is the longest portion and extends from the cell surface outside the body. See this filament, this is the filament, it is extend outside. It is extending outside from the bacterial, and second one is hook, and this is basal body. And last thing we are going to see is besides flagella, what will be the filament? Filament will be there, no? So beside flagella, in flagella only it is having these three parts. So beside flagella we have pillae and fimbriae. So what is pillae and fimbriae? This is flagella. So this is flagella and this is fimbriae and pillae. So fimbriae is what like structures? It is giving what? Pillae. It is in tubular structure. Tubular structure, it is extending the polar end. And this is sprouting out. Pillae is sprouting out. We are seeing, will be seen in the your NCERT books. Pillae and fimbriae also surface structure of the bacteria but do not plays an role of motility. This fimbriae and pillae is an bacteria but it does not gives the motile function. Okay? And the pillae or elongated tubular structure made of specialized protein. This pillae is what? It is an extend towards the structure and it will be consist of full of what? Protein. In fimbriae, the fimbriae are smaller bristle-like fiber sprouting. What is sprouting? What is sprouting? Sprouting வெடிக்கிறதா? முளைக்கிறது. இப்போ for example பீஸ்லாம் வச்சுருக்கோம் நம்ம. பீஸோ வேறு எதுனா நம்ம பட்டாணி அந்த மாதிரி எதுனா நம்ம வச்சுருக்கோம். அதான் பீஸ் தான் நான் சொல்லியிருக்கேன். ஸோ இப்போ அதெல்லாம் நம்ம என்ன பண்ணுறோம்? முளை கட்டின பயிர் சொல்லியிருக்காங்களா? தெரியுமா? ஸோ அப்போ நைட்டு தண்ணியில் ஊற வச்சுட்டு அதை என்ன பண்ணுவாங்க? ஒரு ஈர துணியில் கட்டி வச்சுருவாங்க. ஸோ அப்போ அது என்ன ஆகும்? நெக்ஸ்ட் டே மார்னிங் வில் சி வாட் வில் சி சம் ஸ்ப்ரவுட்டிங்கில் ஒரு ஒயிட் கலரில் ஒன்று வெளியில் வந்திருக்கும். ஸோ அதுதான் ஸ்ப்ரவுட்டிங். ஸோ அந்த மாதிரியே வந்து என்ன இருக்கா? fimbriae இருக்கு அப்படின்னு சொல்லி சொல்றாங்க. In some bacteria they are known to help attach the bacteria to rock in stream and also the host tissues. So according to bacteria நம்ம பார்க்குறோம் அப்படின்னா maximum bacteria வெளியில இந்த rock stream கிட்டலாம் இருக்கும். ஸோ ஈஸியாக இந்த fimbriae எல்லாம் இருக்குது அப்படின்னா ஈஸியாக it can என்ன பண்ணும்? அட்டாச் ஆகிடும். போய் அங்கே இப்போ ஒரு முள் புதர்லாம் இருக்குது. ஸோ அது கூட என்ன ஆகும்? ஈஸியாக போய் அட்டாச் ஆகிக்கும், வராது இல்லையா? ஸோ அதுதான் வந்து இது ஒரு ஹோஸ்டிங் பிளேஸாக வந்து இது கன்வெர்ட் ஆகிடும் அப்படின்னு சொல்கிறாங்க. ஸோ தீஸ் ஆர் ஆல் த திங்ஸ் in prokaryotic cells. So next we are going to see about ribosomes and inclusion bodies. So ribosomes and inclusion bodies we are going to see tomorrow morning at the same time 8:30. So we will be starting ribosomes and the inclusion bodies. So far any doubts you can ask me tomorrow and recall the subjects. So before entering into the class we will recall this day about the subjects. Okay children, thank you.